to pop in before this session with a couple of housekeeping things. Wanted to share some photos from Rock the Run this morning. Again, really appreciate you guys coming out. I also wanted to share this update, which is really exciting. So that's my slide of choice for excitement. So when we did Rock the Run a few years ago for the first time, I think we had set a goal to raise $10,000, and we were really excited when we hit it. And then last year, we set a goal for, I don't even know, I should have looked it up. I think it was like 30, and we got to maybe 24. This year, we set a goal for $50,000, and we are right now this afternoon at $46,000. So thank you. And that, when you go on the Rock the Run page, it doesn't reflect that total because we have like satellite events. We have families, patients and their caregivers and their families doing both memorial walks and runs. And they tell us that when they see these pictures with their doctors, with the researchers, the people that they follow from afar on Twitter out there running, that it means so much to them. So I want to say thank you for that. Thank you. I really appreciate that. There is a QR code on your tables if you still would like to donate. We have that little gap that we're still trying to close. So thank you for that. And I also want to thank, I'm not sure if anybody actually is here from ASI at this point. Thank you. Because ASI has donated and supported Rock the Run before we made any money with it. And that, I think those totals really show how your contribution is multiplied. So thank you very much for that. All right. Brian, are you introducing our next? Okay, Brian's going to let you guys introduce yourself. I'm just going to clear out.
All right. Hi, welcome back. So our next session is the rapid oral abstract presentations, and Dr. Powles and myself will be the moderator for the event. I guess why don't we just get started? Let's go. The first speaker is Nicholas Salgia from Roswell Park.
Thank you for the invitation. So I was talking to Dr. Surzan yesterday at the posters and promised him an exciting and fun conversation on this one. I'm going to try to live up to that and actually see if I can get this thing to work. So we've heard quite a bit about sarcomatoid RCC. I'm going to just skip the background and jump right into it. Our group at Roswell Park, I can actually stand right here with the microphone. That's probably a little bit better. Is much more interested, or very much interested in the immune responsiveness, the paradoxical immune responsiveness that we see in these sarcomatoid RCC tumors. This is under the tutelage and mentorship of Dr. Jason Muhich, who is a brilliant tumor immunologist sitting right over there. If you haven't met him, definitely go up and talk to him. And to do this, we use single cell RNA sequencing on 18 clear cell tumors. 10 with a 10 with a sarcomatoid compartment that I'll refer to as sarcomatoid RCCs, and 8 control clear cell onlys. This is what it looks like following dimensionality reduction and clustering in Tisney space. And I'll give away the punchline right here. There's really only one major cell population that is different between these two subtypes, and that's the plasma cells. So let me take a few steps back and take a look at this a little bit more. So based on some really exciting preliminary data I don't have time to talk about today, we started by looking at the CD4s and the CD8s and the T cell populations. This, in this case, is the CD8 population. I'll give you the caveat that what we see in the CD4s is very similar as well. So this is after isolating them and reducing their dimensions, performing differential gene expression between the sarcomatoid clones and the clear cell clones from the T cells. We see that CXCL13 is by far and away the most dramatically enriched gene in the sarcomatoid RCC-derived T cells. In fact, when you actually look at this in the relative proportion of T cells, there's over 40% of them are expressing CXCL13 in the context of the sarcomatoid microenvironment, compared to only about 20% in the clear cell microenvironment. And we went ahead and corroborated this enrichment of CXCL13 in multiple bulk data sets comparing sarcomatoid tumors to epithelioid tumors, including a real-world data set from the City of Hope through a collaboration with Monty Pal and the others down there. So I'm in an immunology program. The first question I get asked whenever I say CXCL13 is, oh my goodness, Nick, did you look at tertiary lymphoid structures? And the answer is, heck yeah, we did. In fact, we used 2 different transcriptomic signatures. First, the canonical 12 chemokine signature, and then on the bottom, the imprint TLS signature from Wolf Friedman's group in their great immunity paper a few years back in kidney cancer. And we showed across these trials where we had access to raw counts or normalized counts matrices at a per patient level, both of these signatures were significantly enriched for in sarcomatoid defined tumors, compared to clear cell or epithelioid tumors. And we have a small cohort of spatial samples as well with known sarcomatoid compartments. And by mapping canonical TLS genes like CD3, CXCL13, CD20, and MZB1 for our plasma cells, we can actually identify a locus that is pathologically confirmed as a TLS in the perisarcomatoid region. So these TLSs are ectopic foci of lymphocyte activation and antigen cross-presentation, and the maturity of them are often defined by the presence of a B cell follicle. So we next wanted to look at the actual B lymphocyte compartment. Again, we used our single cell cohort to do this. This is what it looks like when we look at our B cells and our plasma cells. On the top are our IgG expressing or IgA or IgM or IgK kappa expressing plasma cells. On the bottom are our CD19 and CD20 B cells, And you can really see on the bottom there's a marked difference between the sarcomatoid-derived clones and the clear cell-derived clones. Again, in fact, when you look at the proportion of plasma cells in these cohorts, over 40% of B lymphocytes in sarcomatoid RCC are plasma cell, compared to less than 4% in the clear cell RCC derived tumors. Again, using some publicly available bulk transcriptomic data. This is from Ziad Bakuni's Nature Comms paper. We actually went into their differential expression analysis and performed gene set enrichment analysis on a number of B cell associated pathways and showed activation and enrichment of B cell activation in plasma cell and immunoglobulin secretory programs in the sarcomatoid RCC microenvironment. And the last thing I'll show you for the sake of time is we're also showing expression or enriched expression of Fc gamma receptor 3A or CD16, which is the mediator of antibody-dependent cellular cytotoxicity in the microenvironment. And for the sake of time, I won't show you, but we're actually really isolating this down to the monocytic lineages within the myeloid compartment rather than the NK cell populations. And I know Vivien from Dr. Braun's group will be talking a little bit more about NK cell dysfunction and advanced RCCs coming up. And so I think there's a nice parallel to draw there. These are the conclusions I just walked you through. This is just one small part of a very multidisciplinary project at Roswell Park. So thank you to all our collaborators, colleagues, and all of you, as well as our funding sources. Thank you.
Fabulous. Was it fun? Roy, would you like to give your presentation? I think it's on tumor evolution of brain-specific tropism in metastatic clear cell renal cancer.
I would love to. Thank you to the organizers for the opportunity to present our work evaluating brain metastases in RCC. So this crowd may be aware, brain metastases occur in approximately 10% of patients with metastatic RCC. Interestingly, the distribution and the cadence of RCC metastases are not entirely random. It seems that the primary tumor and the biology of the primary tumor can influence this behavior. From a brain metastases perspective, pan-cancer studies evaluating lung cancer, melanoma, a few RCC samples have implicated the mTOR pathway and deletion in chromosome 9p, encoding for CDKN2A or containing CDKN2A as a common mechanism in the development of brain metastases. However, many of these studies were limited in the number of RCC-specific samples, and so we wanted to explore the molecular landscape of brain metastases in RCC specifically. In our study, we identified patients at JHU underwent a craniectomy for RCC metastasis. It's an interesting projection there. Panel and DNA-based sequencing and RNA sequencing were performed on all resected brain metastases for the cohort, and non-brain metastases in primary tumors when available. We compared somatic mutations, copy number alterations, and gene expression between the 3 tumor sites. Projecting a little bit strangely there, but histology is 85% clear cell RCC for the cohort. And the bottom panel there is showing that most patients, approximately 60%, had more than one sample. So a brain metastasis and a primary tumor or a non-brain metastases. From a somatic mutation standpoint, we saw that there was a relative enrichment of TP53 and PTEN mutations in metastases, with the latter, PTEN, seemingly enriched in brain metastases, although this did not reach statistical significance in this cohort. From a copy number alteration standpoint, we saw an enrichment for chromosome 9p21 deletions in brain metastases compared to primary tumors. And from a copy number gain standpoint, we saw gains in chromosome 3q and chromosome 20q. Differential expression analyses comparing brain metastases, non-brain metastases, and primary tumors revealed a number of differentially expressed genes between these groups. And gene set enrichment analysis showed some interesting patterns. When looking at metastases, so both brain metastases and non-brain metastases relative to primary tumors, we saw enrichment for cellular proliferation pathways in the epithelial to mesenchymal transition. In brain metastases, they were specifically enriched for mTOR, glycolysis, mixed signaling, hypoxia, and protein secretion hallmark pathways. Alternatively, angiogenesis, inflammatory response, and interferon gamma and TGF-beta were downregulated in brain metastases when compared to non-brain metastases as well as primary tumors. We built upon this with cellular deconvolution using CyberSort, shown here in this heat map, the entire LM22 signature. But a few of the cell types that I choose to highlight are CD8 and M1 macrophages, which were down in brain metastases relative to primary tumors and non-brain metastases, whereas mast cells, M2 macrophages, and neutrophils seem to be enriched in brain metastases versus primary tumors. So in conclusion, we saw PTEN mutations and CDKN2A deletions, which seem to be enriched in RCC brain mets, consistent with prior work. mTOR and mixed signaling was specifically upregulated in brain mets relative to non-brain mets and primary tumors. And brain mets demonstrated an immunosuppressive tumor microenvironment phenotype. I'd like to thank my collaborators and my mentors who contributed to this work, the patients, and some of the funding sources there. Thank you all.
Thank you for that interesting presentation. The next presentation is Impact of Latino Ethnicity on the Gut Microbiome Compositions of Patients with Metastatic Renal Cell Cancer, presented by Regina Barragan-Carrillo from City of Hope.
Thank you very much. My name is Regina Barragan-Carrillo. I am a medical oncologist and a postdoctoral fellow at the City of Hope Comprehensive Cancer Center in Duarte, California. Today I'll be presenting our work on impact of Latino ethnicity on the gut microbiome composition of patients with metastatic renal cell carcinoma. Sorry about it. So overall, the Latino population in the United States represent the largest minority group. Overall, there are about 60 million inhabitants in the country, representing somewhere in between 19 and 20% of the whole nation's populace. The first cause of death of Latinos living in the United States overall is cancer, and metastatic renal cell carcinoma is not an exception to this. We have data pointing out that patients who have been diagnosed with metastatic renal cell carcinoma who receive frontline treatment with immune checkpoint inhibition present shorter progression-free survival as well as higher odds for both disease progression and death. Recent studies have linked that the composition of the gut microbiome with outcomes with immune checkpoint inhibition. Furthermore, several studies on the gut microbiota have demonstrated a distinct microbiome profile among different ethnic and racial groups. With this in mind, we aim to investigate the differences in the gut microbiome composition between Latino and non-Latino patients with mRCC. We examined samples from 2 phase 1 single-center randomized controlled trials that included treatment-naive patients with mRCC, and patients provided a stool sample with the OmniGene gut kit at baseline. Whole metagenome sequencing was performed on stool specimens collected. Overall, we collected samples from 27 Latino patients and 32 non-Latino patients. The median age of the whole cohort was 65 years. Most patients, 85%, had clear cell histology, and 15% had sarcomatoid component. Most had intermediate risk IMDC disease. When looking at response to treatment, we noted a statistically significant difference in the disease control rates between Latino and non-Latino patients, favoring the Latino patients 100% versus 77%. Looking at the microbiome analysis, we noted that there were no statistically significant differences in the Firmicutes to Bacteroidetes ratio. Similarly, we did not observe any differences in the alpha and beta bacterial diversity. Something very interesting that we did observe is that the Yancom BC analysis showed that there were 14 bacterial species which were enriched within the Latino cohort and 3 that were depleted. Namely, 3 Roswellia species, Roswellia feces, Roswellia hominis, and Roswellia inolivorans. I would like to highlight the fact that Roswellia is one of those bacterial species that have been classified as butyrate producing, which makes the findings quite interesting. Furthermore, we also noted an increase in the Bacterium rectali species, Walteria intestinalis, and Bacteroides merde. Within the non-Latino patients, we note an enrichment in a Methylobacterium bacterial species, which was not classified. With this, we came to the conclusion that Latino patients with mRCC showed an increased abundance of Roswell species, Bacterium rectale, Walteria intestinalis, and Parabacteroides merde in the gut microbiome analysis. Most of these species have been previously linked to favorable clinical outcomes with immune checkpoint inhibitors. We overall observed better clinical outcomes in Latino patients in our series. Nevertheless, we want to highlight the fact that there is still a need for further examination of this hypothesis in a larger series. We speculate that the higher rate of bacterial species linked to ICI responsiveness might be part of the driver of these better outcomes that we notice. Clinical trials related to microbiome should potentially account for baseline differences in ethnicity, and I want to finish by mentioning the fact that microbiome modulation presents a unique and cost-effective strategy to aid in reducing the disparity that the Latino patients with cancer face, highlighting its potential value in improving outcomes for minority populations. Thank you for your attention.
That was great. We've got time for 6 minutes of questions. Any takers from the audience before I kick off? Far away.
Thank you. Questions for Nick. First, I'm curious, in the tumors where you did spatial analysis, what percentage of tumors actually had the TLS structures? And then kind of on a related note, do you think that your gene expression signatures are reflecting purely the presence of TLS, or could it be less mature lymphoid aggregates present within the tumor?
Cool. Fantastic questions. For the first one, I'm curious as well. So we really have a small cohort of spatial seq right now that we're currently expanding. We've shown it in this one for sure, confidently. I actually haven't even looked at the other three because this is the first one I analyzed, and it was a perfect picture, right? What we're doing is using multispectral IF to actually confirm these at the proteomic level right now, as well as just H&E annotations via pathologists for the presence of them. So I'll get back to you with an answer on that first question, hopefully in the very near future. The second question, I'm sorry, remind me. I have a memory of a fly, clearly.
Well, so you might then not be able to answer the second either.
Lymphoid aggregates, right?
Are the expression signatures reflecting purely the TLS, or could it be less mature lymphoid aggregates?
Great question. So Carrie Jansen's here, right? Carrie put that beautiful Nature paper together a few years ago with Hayden Kissick's group looking at these lymphoid aggregates in the intratumoral space, really necessary as a second step for T cell activation in the sarcomatoid or in the RCC microenvironment, right? We aren't going to rule that out as a mechanism of antigen cross presentation or lymphocyte activation and unique to SARC or enriched in SARC. But both of these signatures that we are applied at the bulk transcriptomic level are validated to be true TLSs. Wolf Friedman's group, that immunity paper led by Maxime Malin, is really fantastic at showing that in a rigorous manner.
Thank you. And then finally, have you seen any correlation between circulating CXCL13 and intratumoral T follicular helper cell?
Fantastic question again. Have not looked. Thank you.
There was a question down here, I think was next, in the green jackets. If we can get the ball over in that direction. Nazar's going to act as, oh, not bad. Excellent. Yeah.
Very much.
Good white receiver.
I was just going to comment to Roy about the metastases and mTOR, that the the gene set enrichment analysis for mTOR is, in my mind, really flawed. And it has nothing to do with mTOR signaling exactly. So you might look at there's a specific variant of MTHDF2 that seems to be an indicator of mTOR activity rather than just random genes that are somehow connected to the pathway. And it would be very, very interesting if there is a link between brain metastases and mTOR. So.
Yeah, that's actually a wonderful comment. I think the hallmark gene sets, which we tend to look at, they all suffer from this issue that you've just described. There are other gene sets which we certainly can look at, and I will take your advice. Thank you.
We've got a question there, far away. Then one at the back, and then we're probably going to have to stop. No, I was in the front first.
Oh, me?
Far away.
Okay. Well, Lisa largely stole my question, but this is for Roy. because, my read of all the genetics suggests that there's hits up and down the PTEN-mTOR pathway. And if I understood your presentation properly, this was sort of specific for PTEN mutations and not other things that might be in the epistatic scenario. And I was just wondering what your thoughts were about that.
That's another great point. So I did look at activating agents in the mTOR pathway, including TSC1, TSC2, and mTOR. One thing I should mention is the sequencing that was done here is panel-based without a matched normal. So there is some challenges in calling this. The biggest signal was for PTEN, which is what I put here when I included all those different mutations. The difference between metastatic sites diminished. Having said that, I think that it is the fact that we're seeing PTEN mutations on the genetic level. We're seeing this in other tumor types as well. There was some single cell data, which I've seen in the abstract form, which is suggesting in the tumor epithelium, there's an enrichment for mTOR. And we're seeing it on the gene expression level, albeit flawed. There may be some signal there. I will also note a very important confounder here is for all patients who undergo brain metastases resection, they all receive systemic steroids as part of standard of care. And I'm not sure how, it's very difficult to correct for that confounder. I haven't thought of a great way to correct for that confounder, but I'm looking at genes that are differentially regulated with steroid use to see what overlap there is there and how much of it is explained by that.
Great, thank you.
We've got a question in the back and then we'll have to stop.
Yeah, I think in similar vein, I think in terms of brain tropism, some suggestions for consideration are, I'm assuming a surgical pathologic analysis has been done and these are truly brain metastases. The reason I bring this up is primary renal hemangioblastomas have been published in the last year, a small series of cases, and all of them are highly enriched for mTOR TSC pathway aberrations. So one suggestion is to also check for biallelic VHL status. Do any of these patients have a germline VHL loss? And is there any chance that any of these tumors could be actually a hemangioblastoma instead of a metastatic renal cell carcinoma? Unlikely, but I think something good to check across. But primary renal hemangioblastomas have now mTOR TSC pathway aberrations, which are like highly implicated. So I think just doing some basic surgical pathology markers, I think might just help out. Thank you.
That's a great point. All of the pathology was reviewed by an in-house pathologist, Dr. Azar-Garban. I'm getting signals, but we can talk after.
Fabulous. Thank you very much. Thank you. We're going to invite Vivien Moritz to the stage, who's going to talk about natural killer cells and their impaired cytotoxicity. And in parallel, if Soki and Shahala can come to the stage for their presentations as well. Oh, a third one too.
Yeah.
Oh, yes.
Salem.
And Seema. And if Seema Salim could come as well, that'd be great also. I'm doing a brilliant job.
Okay, you may start. Yes.
Fabulous, thank you.
All right, thank you.
Welcome.
So, hello everyone. Today I'm going to be discussing natural killer cells and their impaired cytotoxicity in advanced renal cell carcinoma. So just for some brief background, natural killer cells are extremely important for anti-tumor immunity within multiple different cancers, including clear cell renal cell carcinoma. However, NK cell function and phenotype is not clearly understood. So we began investigating this question by asking how NK cell function and phenotype evolve with advancing disease stage. To do this, we utilized single-cell sequencing data from 13 treatment-naive patients from an existing data set our lab has previously reported on. The NK cell population from this data set resulted in over 21,000 high-quality NK cells. So, wanting to investigate the evolution of NK cell composition and phenotype within disease stage, we initially viewed that samples with more advanced disease states had a significantly lower proportion of NK cells versus total immune cells compared to localized RCC and normal tissue. They're advancing down here, but I'm not sure. Oh, okay, perfect. So to do this, we utilized single cell sequencing. I already went over that. Okay. Next, we wanted to take a deep dive into this NK cell population. Clustering analysis revealed 6 distinct subsets. These clusters were then annotated using canonical markers for CD56 dim and CD56 bright genes. While most clusters expressed these more dim-like markers, cluster 0 expressed, you can see there at FCGR3A, there was a marked lack of the expression of the CD16 gene, which cued us into its more bright-like tendencies. When analyzing these clusters across disease states, we also noticed that the C0 cluster was significantly enriched in advanced RCC. So, wanting to further investigate why, we performed differential gene expression analysis, and this analysis revealed an increase in the expression of markers for tissue residency, such as ITGA1, and CD9, and decreased expression of cytotoxic genes, such as granzymes and perforin. So we wanted to be able to validate this in a more generalizable way, so we turned to the TCGA clear cell cohort. We created gene expression signature to interrogate bulk RNA sequencing data from this cohort, and the advanced RCC samples had a considerably higher gene signature score, showing us this phenotype was enriched in advanced RCC, and we had validated our results in an external cohort. So our RNA sequencing data suggests that this C0 phenotype is defined by this decrease in cytotoxicity, but we wanted to be able to confirm this. So we turned to in vitro analysis and isolated NK cells from the blood of patients with advanced ccRCC, and then co-cultured these with target K562 cells. After co-culture, our CD49A positive, CD9 positive cells, which are more representative of our C0 population, had significantly decreased cytotoxicity compared to our CD49 negative, CD9 negative population. So this validated our findings, and we were able to observe this cytotoxicity. Just for some brief conclusions to wrap up everything, there's clearly this dysfunctional NK cell phenotype, which is enriched in advanced ccRCC, and we were able to generalize this to a broader population. And we were able to also validate these findings in vitro, as these impaired NK cells did have decreased cytotoxicity. So thank you to all my fellow co-authors for all your assistance, and funders and donors. And thank you so much to Dr. David Braun for giving me this opportunity.
Thank you, Vivien, for the presentation. Next, we have development of an ex vivo patient-derived tumor model to assess the tumor microenvironment in renal cell carcinoma, presented by Soki Kashima from Yale.
Hi, everyone. Thank you for having me at this wonderful meeting. So I'm Soki Kashima, and a postdoc in Dr. David Braun Laboratory at Yale. Today, I'd like to talk about development and ex vivo patient-derived tumor model, PDTM, to assess the tumor microenvironment in renal cell carcinoma. So first of all, we are working on the establishment on the cancer immunotherapy model. The question is, how can we model the cancer immunotherapy clinically? We need to preserve the tumor microenvironment like structure and composition and function. So I'd like to show our data for these three aspects today. For the first is for structure. So we took the air-liquid interface system for this model, and we culture some tumor chunks in this system. When we look at the histology, PDTM preserved the feature of the clear cell carcinoma, and that also preserved the FACS8 tumor cells and CD45 immune cells, CD8 T cells, and CD163 M2-like macrophage. So we can see beautiful structure on this PDTM system. So next step is analysis of the cell composition using multicolor flow cytometry. Here is a parent tumor on the day 1 and day 2 of the PDTM tumor. When we look at the broad population, so we can see ENPP3 positive tumor cells and CD45 pan immune cells, CD3 T cells, and CD11B myeloid cells. So and then, so when we look deeply into the more detailed and the T cell phenotype, and see whether T cell phenotype are preserved, we can find CD4 T cells and CD8 T cells, CD25 regulatory T cells, and PD1 positive CD8 T cells. So finally, when we look at the myeloid cells, and what myeloid cell types are preserved. So we can see tumor-associated macrophage and M2-like macrophages. So we can see these all of type of cells in this PDTM system. So that makes the functional test possible. The final slide is for functional test. When we look at the immune activation marker on the T cells and the cytokine production using the ELISA. As you can see, the anti-PD-1 antibody clearly increased the frequency of the CD25 and the LAG3 positive CD8 T cells. That means the anti-PD-1 antibody makes the CD8 T cell be activated in this ex vivo PDTM model. And interestingly, when we look at the cytokine production, anti-PD-1 antibody makes the PDTM system produce the much higher interferon gamma. So our data showed that PDTM system is a helpful tool to test the novel drugs in the future and a good tool to predict the efficacy. I'd like to thank the patients who participated in this study, as well as their family, and I really appreciate my mentor, Dr. Brown, and my lab member, including Catherine, Vivien, and Lucy. Catherine and Vivien is really working on making the beautiful tumor bank, and Lucy is helped this project, especially ELISA. And VOD funding is supporting our project. Thank you so much.
Thank you, Soki. I'm going to invite Shala Bari, please, who's going to come and talk about fasting-mediated gut microbiome modulation, improving response rates to immune checkpoint inhibition.
Thank you so much for the opportunity. The topic for current discussion is how fasting mediates the change in microbial metabolomics to improve immune checkpoint therapy responses in renal cell cancer. Let me take you to the background that forms the pillar of this study. Our first preliminary data came from the Conquer Cancer Foundation YIA study, where we sought to evaluate the role of microbial metabolome in predicting resistance to immune checkpoint therapy. We prospectively accrued 100 metastatic treatment-naive clear cell patients, and we noted significant differential abundance of microbial metabolites, specifically indole acetic acid and indole acetaldehyde, to be differentially abundant in patients who were resistant to immune checkpoint therapy. These metabolites are already known to be immunosuppressive. They upregulate the immunosuppressive Tregs and MDSCs. They decrease the effector CD8 T cell function. They increase the invasiveness, the migration, and the proliferation of the tumor. Previously, we had already noted a similar pattern in metastatic head and neck cancer patients who were treated with pembrolizumab. So the next logical question was, how do you modulate the microbial metabolome to improve ICB responses? We hypothesized that time-restricted eating could modulate the microbial metabolome and improve immunotherapy responses. And indeed, we found that in a non-randomized pilot phase 1 study in metastatic head and neck cancer patients, we found a significant signal of efficacy of fasting. With patients who fasted, their median progression-free survival was not reached versus 4.5 months for patients who did not fast, with a hazard ratio of 0.2. More importantly, as expected in our hypothesis, fasting led to a decrease in indole acetic acid and indole acetaldehyde. That led to the foundation of our current hypothesis and our current study. Fasting would favorably modulate the microbial metabolome and improve responses to immune checkpoint therapy in renal cell cancer. And we demonstrated this in preclinical kidney cancer models utilizing RENCA and LVRCC cell lines that we borrowed from Dr. Hakimi's group. Our first experiment was conducted in SPF tumor-bearing mice that was treated with anti-PD-1 and cabozantinib. One group was randomized to 14 hours of daytime fasting, while the other group was exposed to ad libitum feeding. And as you see in the growth curve, mice that fasted, there was significant reduction in the tumor growth, improvement in the progression-free and overall survival as compared to the control mice. The next question was to tease out the mechanism. For this, we conducted 2 experiments. The first experiment, though not shown here, was an exact replica of the prior slide, but now conducted in germ-free mice that lacks any microbiome. And interestingly, there was no difference in the tumor growth, progression-free survival, or overall survival in germ-free mice that was fasting versus not fasting. We then went a step further and we transplanted the microbiome of SPF fasting mice into a group of germ-free mice, while another group received the microbiota of a non-fasting mice. These mice did not fast, but what you would note in this growth curve is mice that were engrafted with a fasting microbiome, their tumor considerably regressed and became non-visible, and this effect was not seen in the mice engrafted with a non-fasting microbiota, thus demonstrating that the change in the gut microbiome drives the effect of fasting and is sufficient and necessary to elicit the effect of fasting. Finally, to demonstrate the effect of the microbial metabolite as the mediator of fasting, we again conducted the same 2 experiments in SPF and germ-free mice. In one group of SPF mice that was treated with anti-PD-1, cabozantinib, as well as fasting, we randomized one group to indole 3-acetic acid, while the other group received placebo. And as you note, mice that received indole 3-acetic acid, they had an exponential tumor growth and lost all effect of fasting. Similarly, in germ-free mice engrafted with a fasting microbiota, treated with 8 doses of anti-PD-1, with regression of the tumor, the mice that received indole 3-acetic acid, they lost all effect of fasting. We therefore conclude that fasting improves immunotherapy responses in kidney cancer preclinical models, and the effect is mediated by the gut microbiome and the microbial metabolome, and it is driven by the microbial metabolome, which is sufficient and necessary to elicit the effect of fasting. The question now is fasting ready for prime time in kidney cancer patients? And my answer is an emphatic yes. And finally, can we modulate the microbial metabolome through small molecules to elicit the same effect of fasting in patients who cannot fast? Thank you so much. And none of this work would have been done without my lab mentors and lab members, as well as my mentors, Dr. Dan George and Dr. Conijo Garcia. Thank you so much.
Thank you, Sawa. Next speaker is Summer Salem.
Yes.
Presenting ferroptosis suppressor protein 1 is a potent and targetable inhibitor of ferroptosis in chromophobe renal cell.
Hey everyone, thank you for the opportunity to present my work today about ferroptosis suppressor protein 1 as a potent and targetable inhibitor of ferroptosis in chromophobe RCC. But before starting, I want to acknowledge and thank everyone at the Hansky Lab, as well as our collaborators and sponsors for making this work possible. So ferroptosis is a reactive oxygen species, or ROS mediated, iron catalyzed, distinct form of cell death. It's different from necroptosis and apoptosis. And its hallmark is lipid peroxidation. And when membrane phospholipid peroxidation occurs, this leads to membrane loss of integrity of the plasma membrane and eventual cell death. And back when ferroptosis was first described more than 10 years ago, GPX4, or glutathione peroxidase 4, was thought to be the only protein that the cells had to protect themselves from ROS species that were floating around in the cell. And glutathione peroxidase 4, as its name suggests, allows the reduced form of glutathione to scavenge lipid peroxides and keep ferroptosis at bay. More recently, another ferroptosis suppressor was described. It's called ferroptosis suppressor protein 1, and it acts by reducing CoQ10 to its reduced active form, which is then able to scavenge lipid peroxides and keep ferroptosis at bay. And as we're going to see later, RSL3 inhibits GPX4 and FSN1 inhibits FSP1, and these are ways to induce ferroptosis in the cell. And our current work will focus on FSP1, which up to this point has not been well studied in chromophobe or in many other cancers, compared to the more classic or canonical pathway, which is the GPX4 glutathione pathway. But as a background, in 2018, our lab found that glutathione levels were extremely elevated in chromophobe RCC compared to matched normal kidney. And in a follow-up paper, we saw that we described that chromophobe RCC was hypersensitive to ferroptotic cell death, both in vitro and in vivo, via the disruption of glutathione homeostasis. And this was true even when compared to clear cell RCC, which was found to be also very sensitive to ferroptosis in a prior paper. So we looked at the cancer therapeutic response portal, which was compiled by the Broad. It includes a panel of 800 cell lines, and it takes their gene expression and compares it to its sensitivity/resistance to pharmacologic compounds. And in this graph, we can see resistance to RSL3, which is again a GPX4 inhibitor. And each dot here represents a gene, and we see that the number one upregulated gene is FSP1, which is basically the second arm, or the more recently described arm of ferroptosis suppression. So when we saw this, we looked at the cancer genome atlas, and we saw that, like GPX4, ferroptosis suppressor protein 1, or FSP1, was also upregulated in chromophobe RCC compared to normal kidney. And more strikingly, out of the more than 30 cancers in the TCGA, or the cancer genome atlas, we see that chromophobe RCC has the second highest FSP1 upregulation compared to its normal organ. So in this case, the kidney, out of these 30 cancers. So we moved on to both genetic and pharmacologic inhibitors of GPX4 and FSP1. And here we're looking at siRNA mediated inhibition. And as you see in magenta and in green, at these levels of inhibition, we did not see much cell death when each gene was inhibited alone. But in purple, we see that when we inhibited these two genes simultaneously, we had almost complete cell death, which was not reversed by the necrostatin 1, which is a necroptosis inhibitor, or ZVAD, which is an apoptosis inhibitor, was completely reversed by ferrostatin 1, which is a ferroptosis inhibitor, indicating that ferroptosis was occurring. We validated these findings in another chromophobe RCC-derived cell line, the RCJT2. And then we moved on to combined pharmacologic inhibition. And in this matrix here, we're looking at RCJT2, which is a chromophobe RCC-derived cell line. On the x-axis, we can see increasing concentrations of RSL3, which is a GPX4 inhibitor. And on the y-axis, we can see increasing concentrations of FSN1, which is an FSP1 inhibitor. And the lighter colors, so yellow, indicate complete cell viability, while the darker colors indicate complete cell death. And we see that at lower concentrations of GPX4 and FSN1, we don't see much cell death. But when combined, we see complete cell death. We validated these findings in a second chromophobe RCC-derived cell line, the OK876 cells. And then we compared these two chromophobe RCC-derived cell lines to 7860, which is a clear cell RCC cell line. And we saw that overall, again, the chromophobe RCC cell lines are more sensitive to ferroptosis than the clear cell RCC cell line 7860. In conclusion, ferroptosis suppressor protein 1, or FSP1, enables resistance to inhibition of GPX4, and thus the induction of ferroptosis via the classical canonical pathway. And FSP1 is upregulated in chromophobe RCC, and that co-targeting FSP1 and GPX4, and so glutathione and CoQ10 homeostasis, is effective at inducing potent ferroptosis in chromophobe RCC.
At this time, we have 8 minutes for question and answer with our second group of presenters.
We've got a question.
Here.
I'm still not good at this. For the final speaker, did I come away with the impression that FSP1 is intrinsically overexpressed in the chromophobe patients versus the clear cell patients?
So when you look at the TCGA data, indeed, FSP1 compared to normal kidney is much higher in chromophobe RCC compared to clear cell RCC, yes. Again, out of the more than 30 cancers in the TCGA or Cancer Genome Atlas, FSP1 exhibits the second highest upregulation in chromophobe. The first one is endometrial cancer.
Okay, thank you.
Hi. A question related to this is question related to the same talk, and so high majority of chromophobe renal cell carcinomas we know are amenable to surgery, and a small subset will actually metastasize or might have sarcomatoid features. And the question I had was, do we know if the pathway is equally implicated in patients who have metastatic chromophobe renal cell carcinoma? And TCGA, is all clinically localized disease. And have you got a chance to check in the patients who have metastatic deposits in those metastatic tumors, is the pathway equally implicated or not?
This is an awesome question, and we haven't gotten to study this at the lab yet, but we're currently in the work, and Joel Shami here is working on basically seeing how the interplay of P53 mutations, which occurs more commonly in metastatic chromophobe, and which also occurs, and also the sarcomatoid dedifferentiation are correlated with ferroptosis sensitivity, because we know from Jesse Woodlacker, a collaborator of ours, that in order for cells to metastasize, they need, through the blood at least, they need to be resistant to ferroptosis, because the blood is rich in oxygen and is a milieu that is very basically good at inducing ferroptosis in cells.
Yes, and that's well taken. I just feel that the canonical pathway changes in chromophobe RCCs from a mutational as well as CNV standpoint tend to alter once tumors acquire either a sarcomatoid or metastatic phenotype. So having some, I think, evidence might be helpful. Thank you.
Thank you. We're 5 minutes over, Tony. Do you want to go very fast?
No, just the last one, maybe for Lisa also and Samer. How far are we from these ferroptosis inducer in clinic?
So up to now, we're working with two collaborators in biotech, one in Utah and one here in Boston. And unfortunately, up till now, there's no good ferroptosis inducer with a good PK or pharmacokinetic profile. So it's still in the works.
Great.
Thank you for our speakers. I would now like to welcome the third group of speakers, Chinmay Jani, Renee Maria, and Soki.
Soki.
And Chinmay Jani will be presenting concordance analysis of tissue and circulating tumor DNA in renal cell carcinoma, insights from a multimodal real-world database.
Hi, everyone. Good afternoon. I'm Chinmay Jani, clinical fellow at University of Miami and Sylvester Cancer Center. And I'm going to talk about a project on concordance analysis of tissue and ctDNA in RCC. This project was done under mentorship of Dr. Anna McKay at UCSD. So going through the background, next generation sequencing of ctDNA can complement tissue NGS. And the most important thing is it's a non-invasive test that can be conducted serially. Its application enhances the assessment of spatial as well as temporal molecular tumor heterogeneity, providing insights not into just progression, but also treatment response, prognosis, and so on and so forth. However, limited studies have been conducted in RCC. So the aim of our study was to investigate molecular alterations detected in circulating and tissue-derived DNA in patients with RCC. So this was a retrospective analysis of de-identified NGS data from patients with RCC that had both tissue as well as ctDNA testing. And we used the Tempus multimodal database for extracting the data. We included the patients with matched sample, and matched samples meant that these samples were collected within 90 days of each other. and diagnosis, and all patients should have diagnosis of RCC. Analysis included socio-demographic and clinical characteristics and selected pathogenic somatic short variants, or PSSV, and copy number variants, so amplification and deletions both, along with 2 copy number losses. And for concordance analysis, we restricted to only 105 genes, which were tested on the ctDNA panel, and further restricted to short variants with the exception of amplifications and CNL detected by, again, both tissue as well as liquid biopsy. For subgroup analysis, we stratified the results based on presence and absence of metastasis as well. So going into our results, on the first figure, you can see that the breakdown of the tissue biopsy site, majority of this tissue were from kidney, almost 50%, followed by bone, and then followed by lung. In figure 2, this is the prevalence of molecular alteration against the most common prevalence. In ctDNA testing, the most common was TP53, followed by VHL. As compared to in solid tissue biopsy results, it was VHL and then others. And figure 3 is kind of our central outline and our main take-home message over here as well. So this is the breakdown of alteration according to assay detection. It includes both solid tissue, liquid, as well as combination. So focusing over here, the pink color shows that these alterations were detected by both solid as well as liquid tissue, so NGS in both. The green color showed that these alterations were only detected through solid tissue biopsy. However, the purple color shows that these were detected only through ctDNA testing. So a good number which were not even detected by solid tissue detection. And then going into figure 4, it just shows more data about concordance. That concordance alteration identified in solid as well as in tissue in the liquid increased for most of the alterations when it was stratified based on metastatic versus non-metastatic cohort. So metastatic cohort had higher concordance when done with both the results. So with that, we would like to conclude that ctDNA profiling is not only along with concordance that we wanted to test, it is complementary to tissue-based NGS in RCC, and it can increase the detection of genomic alterations. Concordance between ctDNA as well as tissue profiling was particularly higher in individuals who had metastasis, suggesting that there is more potential utility in advanced stages of RCC. And therefore, further research is warranted to elucidate how longitudinal ctDNA analysis can delineate biomarkers of response and resistance at both the mutation as well as at the ctDNA fraction levels. And understanding these dynamics will not only offer valuable insights, will help us understand how to utilize this testing in disease progression, in treatment outcomes, and give personalized treatment to all the patients with RCC. Thank you.
We're going to invite Renee Salabi to discuss intermediate endpoints for overall survival from an IMDC cohort.
Good afternoon, everyone. Thank you, KidneyCAN and the Abstract Committee for this amazing conference and the opportunity to present this work on behalf of my co-authors. As everyone here knows, since 2018, the approval of different ICI-based combination has shifted the paradigm of treatment in metastatic RCC, and that led to increased survival for patients in that space. Today, we have a median OS of 53 months in the ITT population of CheckMate 214. In real-world data, we have 18 months OS rates that are reaching 90% with Nivo-AP and 83% in ICI and VEGF-TKI combinations. And with this increased survival, sorry about the slides, we have a new set of challenges. So phase 3 trials that are leading to FDA approval now necessitate longer follow-ups, larger number of events, and also higher associated costs. Our aim was to determine whether shorter intermediate endpoints that we'll define later are associated with OS in patients receiving first-line based ICI therapy. To do that, we studied patients from the IMDC, and we selected patients who received contemporary first-line ICI-based therapy from 2013 to 2023. The endpoints we studied were TTF, or time to treatment failure, which was defined from ICI start to drug cessation, TTNT, that's below, not very clear here, defined from ICI start to the initiation of the second line, and objective response that was assessed by trained investigators as CR or PR. And we included 1,667 patients in our analysis. As we may expect, median age was 63 years old. Our population was mostly constituted of males. patients who had clear cell RCC versus non-clear cell RCC, and around 45% of patients who had intermediate IMT-CRS group. In terms of presence or absence of liver, brain, and bone met, our population was pretty balanced with 50-50. Nivo-Epi was a combination that was the most frequently received by our patients, followed by Pembro-Axi and other combinations. Now, our first question was, do these intermediate endpoints associate with OS at any time point and at individual patient level data? And to evaluate this, we performed a candle toss correlation with OS. And just for you to know, so a correlation that's superior to 0.49 is considered strong. And here we see that TTF has a 0.49 correlation with OS, while TTNT has a much stronger correlation with OS with 0.67. And these numbers are consistent across all subgroups, and the strongest in the ICI plus TKI arm. We also performed a landmark analysis to answer the question, does a certain intermediate endpoint at 6 months associate with long-term OS? And here we can see that patients who did not have an objective response at 6 months did worse in terms of OS. And this again was consistent across all subgroups. And we see here the highest hazard ratio in the dual ICI subgroup. And that does make sense because we know that patients who respond initially to Nivo-Epi have a durable response and do way better in terms of OS. And an easier way to visualize that is on the Kaplan-Meier curves. And you can see here the patient who did not have a response to first-line therapy did way worse in terms of OS, and they are depicted here in red. To conclude, TTNT has the strongest association with OS. It is potentially a clinically meaningful intermediate endpoint that we would like to evaluate further. Our study has some limitations. It is retrospective, and it has real-world data, which leads to a certain subjectivity from the providers. Future directions include collaborations and data from prospective clinical trials. I would like to thank my PIs who mentored me on this project, Dr. Schwery, Dr. Hang, Dr. Z, and Dr. Brown, patients and their families worldwide, my co-authors, my friend at Dana-Farber and Yale, and everyone for your attention. Please feel free to reach out if you can read my email here. Thank you.
Thank you, Renee. Next is a phase 1 study of fianlimab in combination with semiplimab in patients with advanced CCRCC presented by Ms. Kim from Seoul National University Hospital.
Yeah. Hello, everyone. I'm Ms. Kim from South Korea. I think I must be only one who flew over more than 10 hours to attend this KCRS24. It's an honor to be here. So, concurrent blockade of LAG3 may enhance the efficacy of anti-PD-1 therapies. Fianlimab blocks LAG3 and MHC2-driven T-cell inhibition, and semiplimab blocks the interactions of PD-1 with PD-L1 and PD-L2. This was an open-label expansion cohort study. Adult patients with advanced or metastatic cleared RCC who had received no more than 2 previous regimens of anti-angiogenic therapy and were either anti-PD-1 naive in cohort 3 and anti-PD-1 experienced with the most recent dose within 3 months prior to screening in cohort 4, received the fianlimab plus semiplimab every 3 weeks for up to 24 months. This study assessed the anti-tumor activity of fianlimab plus semiplimab combination therapy in patients with cleared RCC, and the primary endpoint was objective response rate per RECIST criteria. This slide shows patient demographics. Over 15 patients, each in cohort 3 and cohort 4, were enrolled. Median age in cohort 3 and cohort 4 was 64 years. A majority of patients in both cohorts had high-risk features. 60% of patients in cohort 3 and 93% of in cohort 4 had received 2 or more prior lines of therapy, including therapies other than anti-angiogenic therapy. Here we show overall response by investigator assessment for the 2 cohorts. In cohort 3, the objective response rate was 20%, this control rate was 80%, and the median PFS was 6 months. In cohort 4, objective response rate was 7%, this control rate was 73%, and the median PFS was 4 months. In cohort 3, duration of response were 4, 7, and 26 months for 3 responders. Discontinuation of treatment was due to progressive disease or adverse events in 2 responders, and the 3rd patient had completed the treatment. In cohort 4, the duration of response in 1 responder was 6 months, and the patient discontinued treatment due to progressive disease. Let's move on to the safety profile. Treatment-related adverse events occurred in 80% in cohort 3 and 60% of patients in cohort 4. Among them, serious treatment-related AEs was reported in 20% in cohort 3 and 0 in cohort 4. The most common treatment-related AE were rash and infusion-related reaction in cohort 3, and fatigue was most common in cohort 4. Overall, it showed unacceptable safety profiles. In conclusions, fianlimab plus cemiplimab combination therapy demonstrated signs of clinical activity with durable responses on patients with anti-PD-1 naive and anti-PD-1 experienced clear RCC with an acceptable safety profile. So these data support the development of fianlimab plus cemiplimab combination therapy for patients with metastatic clear RCC. As mentioned by Professor Atkins in immunotherapy session today, there are some ongoing trials of anti-LAG-3 and anti-PD-1 in the first-line setting. So we are eagerly awaiting those results of trials. Yeah, I'd like to thank the patients and their families and all investigators. Yeah, thank you.
We've got maybe time for 1 or 2 questions at the most. Can I start with the last presentation with LAG-3? It was LAG-3. It was in combination with immune checkpoint inhibition. Yeah, and the responses weren't great. Like it wasn't 40%. And one of the populations was immune checkpoint inhibition naive. Would you have expected, immune checkpoint inhibition naive population, you'd expect response rate of 30% maybe? And you were less than that, and that was without the LAG-3. Do you have any evidence that it's doing anything?
Yes, although there's no significant numerical difference between the efficacy data of this study and the nivolumab monotherapy data in pre-treated RCC, but I think direct comparison is very difficult because this study is a phase 1 study with a very small number of RCC patients, and they are very heavily treated. So we cannot draw some robust conclusions. But I think the significance of this study lies in generating evidence that support the need for larger scale studies.
I mean, the toxicity is also, there was 3 cases, I'm not sure. Certainly there is some activity, but I don't know. But one question, did you have by any chance any data on single agent LAG-3 in renal cell, like from your phase 1, even 1 to 2 patients?
I hope I can answer the question, but I don't know the exact safety profile of the anti-LAG-3 monotherapy.
I have a question for Chinmay. Can I ask? Yeah. So Chinmay, I think you showed some interesting data and I think a nice talk. And I think, are you surprised by your VHL and P53 findings? Like I think in metastases, it seems VHL is higher, and also P53 is getting picked up more in metastases, but there's some difference in between the tissue versus CTD. And I'm wondering how much of this might be technologic compared to real tumor biology. Are you able to comment?
I guess the biggest surprise was about concordance. We were expecting that the concordance itself will be way higher, but what we were able to see was it's more kind of complementary. TP53 was more picked up in the ctDNA. And to your main question, whether the technique is different. It is true because ctDNA was 105 genes assay only, whereas the tissue was more than 400 gene assay. And we only checked the concordance analysis on those 105 genes. So the other genes which are not picked up in liquid biopsy would still be missed as well. So therefore, like solid and liquid would have that difference too. Regarding whether there's tumor heterogeneity being observed, that's a great question and probably for future studies as well. But knowing that RCC has way different temporal as well as spatial heterogeneity as well from where the tissue is taken, it shows a lot of different data as well. So I guess both of all those things based on that.
Also, the identity of the mutations might be important as well within those genes itself. Thank you.
We're going to go with the last question.
Just to follow up on one question, I think have you considered the possibility that these are, derived maybe from hematopoietic cells that might be, or a process of CHIP or that is present in these patients? Because I think we've seen it before, P53 being more, prevalent in the circulating DNA, but then, in the tumors, the prevalence of P53 mutation is relatively very low in RCC. So I would be cautious to really assign those mutations to the kidney cancer.
We do have one more question online, and I think we're going to conclude the session with that question. So this is for Renee Salabi. What was the median overall survival for the entire cohort? And for all, what median overall survival do you quote? Currently understanding that the real-world data for patient is generally worse compared to clinical trial patients.
Thank you for that question. I should have clarified. I apologize. So we have a pretty variable follow-up for all patients and a pretty short OS here in this real-world data compared to trials because we don't have a lot of follow-up, which is why we had decided to do the only 6-month landmark analysis. And I believe that's where the question is coming from. And yes, patients in the real world tend to have shorter overall survival and outcomes, which is why it was interesting for us to look at the real world data here. Thank you.
Conclude for our speakers. Thank you. We will now break for 15 minutes, about 15 minutes, and please come back at 4 p.m. Thank you.
Thank you very much indeed.